Our sections today are going to cover sections 3.3 and then 3.5 to 3.8, which are cellular organelles. You're going to notice with each of these slides the function, location, and structure of each of these different things. So it's going to be important to note that in your notes, what it does, where it is, and what does it look like, and what is it made out of. So we're going to start with the outside and then move inward. The first one here is a cell membrane. Sometimes this is also called the plasma membrane. It's going to, as its function, it's going to control what enters and leaves the cell. It also serves as the boundary between the cell and the outside world. As far as location goes, obviously it's going to surround the outside of the cell, right? It's the boundary between the cell and the outside world. Structure-wise, we talked about this chemical in our last unit. It's made out of phospholipids. It's a bilayer, which you can see down here. Okay, so you've got the hydrophilic heads facing the outside with the water, and the inside are the hydrophobic tails facing each other. So it's this phospholipid bilayer. And so the last thing I'm going to note on every one of these slides is what kind of cells do you find this in? And of course, if this is in every living cell, we're going to need it to have a boundary between the cell and the outside world. So all living cells, prokaryotes and eukaryotes, have a cell membrane. Next to that, if a cell has this, we're going to see cell walls. So not every cell will have a cell wall. The function of a cell wall is to provide support and protection for the cell. It's going to make the cell rigid and stiff and inflexible, so it should stay pretty sturdy. Location-wise, if a cell has it, it is outside of the cell membrane, so this would be the outermost structure if a cell is going to have it. Structure-wise, at least for plant cells, it's made out of cellulose. We talked about cellulose in our last unit, right? That is a polysaccharide used for structural support. So what cells do you find this in? These are in plant cells, and then some bacteria will also have a cell wall, but theirs is made out of peptidoglycan and not cellulose. So it's found in plant cells and some bacteria. Working our way inwards now, our next one is cytoplasm. So this is going to be the fluid inside of the cell. It's going to be where the organelles are located. That's where they're floating in. That's also the fluid in which the reactions occur. We talked about in our last unit how a lot of reactions have to happen with things dissolved in water. So cytoplasm is not pure water, but it's mostly water. Location-wise, I just said it fills the inside of the cell. In the structure, it is a jelly-like substance. It's mostly water, but it's not entirely water, so things are able to still dissolve in it. And of course, since it's filling the inside of a cell, this is going to be in every living thing. Prokaryote, eukaryote, everything's got cytoplasm. Working our way to the middle of the cell now, our next structure that we'll look at is a nucleus. The function here, it serves as the control center of the cell, sometimes what you'll hear it called. It's where all the genetic material is held, so it contains and holds the chromosomes. Here you can see the nucleus inside of the cell. It's in the middle of the cell typically, unless you have some sort of very specialized cell. The nucleus is usually in the center or very close to the center. Structure-wise, it is surrounded by a nuclear membrane. So when we talked about before the membrane-bound organelles, a nucleus is a membrane-bound organelle. Right? It's surrounded by membrane. So membrane is, once again, phospholipid. So this outer boundary of it here, okay, is made out of phospholipids, right? So a phospholipid layer there. This is only going to be found in eukaryotes. So plant cells, animal cells, protists, amoebas, things like that, okay? It's going to be only in eukaryotes, because we talked about in the last section how eukaryotes have membrane-bound organelles, prokaryotes do not. So this is only going to be inside of eukaryotes. Continuing on with the nucleus, the outside of it, that membrane here, has a special name. It is called the nuclear membrane, or sometimes called the nuclear envelope. Okay, so function-wise, it serves to protect the nucleus, so everything inside of it. And then location-wise, it's going to surround the nucleus. It's made out of membrane, right? It is a double phospholipid bilayer. So structure here, it's a double phospholipid bilayer. So it's kind of interesting. It's not just a single layer, it's actually two double layers. Okay, so it's a double bilayer. This is obviously only going to be, once again, in eukaryotes because we're talking about a nucleus. It's going to be animal cells, plant cells, protists, amoebas, things like that, but it's got to be in eukaryotes. Our last nuclear structure is something called the nucleolus. I've heard this called the nucleolus, but I generally say nucleolus. It's going to be these dark spots inside of the cell. It's usually these dark round bodies inside of the nucleus. It's located within the nucleus, it's dark and round, and its function, it contains the genetic information to make ribosomes, and it's also where a lot of ribosome assembly occurs. Okay, so this is where that's occurring. This is obviously, once again, only in eukaryotes because it is inside of a nucleus. So plants, animals, protists, amoebas. The next one here is called mitochondria. Many of you have probably heard of this one before, maybe in middle school. Function-wise, it's going to release the energy stored in food. We talked about how the energy chemical is ATP. It's the energy currency, right? So this is going to make a lot of ATP. Perhaps you had a teacher in the past call the mitochondria the powerhouse of the cell. It makes the energy. So what is the actual energy? Well, it's ATP, right? Adenosine triphosphate. It's located inside of the cytoplasm. Cells will have generally one mitochondria, though if a cell is something that needs to have a lot of energy, you might have lots of mitochondria. So like muscle cells that require a lot of energy might have more than one of these in there. Structure-wise, it is once again made out of membrane, so it's made out of phospholipids. And you can see here in the middle these folds that are called cristae. These are where the reactions actually happen. They're on these cristae. That's where the energy is being produced. These are only going to be in eukaryotes, so plant cells, animal cells. Okay, sort of a cool biological theory is that mitochondria were once their own separate cells, and the theory for that stems from the fact that mitochondria have their own DNA that is separate from the rest of the cell, separate from the nuclear DNA. So it's theorized that perhaps these were once their own organism that got enveloped inside of a cell, and then you had a symbiotic relationship happening where the mitochondria provided a lot of energy for that cell, and the cell provided a safe place for it to stick around and thrive. So perhaps that was the origin of eukaryotic cells and mitochondria, where they might have come from. At least that's the theory. The next one here is called endoplasmic reticulum. So there's two types. There's smooth endoplasmic reticulum and rough endoplasmic reticulum. Typically, I'll just call this ER, smooth ER, or rough ER. Smooth ER has no ribosomes. Rough ER has ribosomes on it. We'll talk about what those do here in just a moment. Function, both of these kind of work to transport material in the cell. However, there are some specific functions for smooth or rough. Rough ER also has a job of making proteins. Smooth ER, its job is to degrade toxins. So location, it's usually in the cytoplasm. It's usually kind of looks like these folds like that. Somebody once described it as looking like a fingerprint to me. I think that seems to make a lot of sense. Structure, it's made out of membrane, so not going to be in prokaryotes, right? This is only going to be eukaryotic. It's made out of phospholipids, and it's folded, right? It's a series of tubes and canals. Smooth ER, kind of an interesting thing about it, right? It degrades toxins, so the cells in your body that do this are usually liver cells, and so liver cells have a decent amount of smooth ER in them to begin with. Obviously, this is only going to be in eukaryotes since it's a membrane-bound organelle. And the last one here for today are ribosomes. Ribosomes are these little tiny dots here and then down here, each of these little dots. Their job is to make proteins. These are very, very important. And as you might be able to guess then, since their job is to make proteins and every living cell needs to have proteins, it's going to be in everything, right? So prokaryotes, eukaryotes, doesn't matter. Every living cell has to have ribosomes to make proteins. It's located either on the endoplasmic reticulum, specifically the rough ER. That's what makes it kind of look like sandpaper and where the rough ER gets its name from. or they are floating around in the cytoplasm. So if they're in a prokaryote doesn't have any ER because it's a membrane-bound organelle. So in a prokaryote, they're just kind of floating around in the cytoplasm. Structure-wise, they are tiny, round, little dark bodies. They look like little dots, and their job is to assemble proteins by reading messages sent by RNA into it. So we'll talk more about that later on, but that's all you need to know for right now. So we'll stop here for today, and we'll pick up the rest of this next time.